Welcome to the Molecular Microbiology Laboratory at Humboldt University in Berlin. Today you will learn a technique to study phenotypic heterogeneity in bacterial response to antibiotic treatment. Antibiotics are the first-line treatment for bacterial infections. However, over time, bacteria have evolved resistance mechanisms to cope with the presence of antibiotics, rendering them unresponsive. Antimicrobial resistance is a serious and growing public health problem worldwide. Resistance renders standard treatments ineffective, leading to unresolved disease and increased risk of spreading infections. In 2019, antimicrobial infections will cause nearly 1.3 million deaths worldwide, more than deaths from HIV or malaria. A fundamental strategy used by bacteria to prepare the population for unexpected changes in their environment is the principle of phenotypic heterogeneity, a process that describes that individual bacteria can behave and respond differently to extrinsic stresses, such as the presence of antibiotics. To study such behaviors, techniques are needed that resolve the bacteria to the single-cell level. Today, we will study the response of the gram-negative bacterium Salmonella, a foodborne gastrointestinal pathogen, to exposure to beta-lactam antibiotics such as penicillin or a newer member of the beta-lactam family, ampicillin. To do so, you will take the role of junior researcher for a day. I know that Dr. Molecular has a special assignment for you today that will allow you to understand the mechanisms of antibiotic resistance and a new technological approach that we are using in our laboratory to study such behavior. Oh, I think she is calling already. Please take the Skype call by clicking on the phone button on the display. She will explain everything further to you.
Hi, I'm so glad I can reach you. We need your help with a food sample. Can you check in the lab to see if the isolated salmonella can be treated with ampicillin? We need to know how the bacteria behave to stop the spread of these pathogenic bacteria. To resolve the behavior of individual salmonella bacteria to ampicillin, you can test their response using our microfluidic device. No worries if you have never used it before, I will show you its basic functions before we perform the experiment together. This microfluidic chip, often called a lab-on-a-chip, is a miniaturized device that integrates multiple functions on a single chip. It is typically millimeter or centimeter in size, and is typically made of elastomeric material, such as polymers or silicon. The chip is designed to control and manipulate small amounts of fluids through its unique design of microchannels and chambers. For our experiment, we will use the so-called mother machine design, which is perfect for tracking the growth of single cells over multiple generations. By mounting this chip under a microscope, we can monitor the bacteria as we expose them to different environmental conditions. To observe the behavior of the bacteria, we first load the bacteria onto the chip using a syringe. We then flush beneficial media, colored green, into the chip to support bacterial growth. Once the bacteria reach the steady exponential growth phase, we can quickly switch to media supplemented with an antibiotic, colored in red, and observe how the bacterial population responds to that antibiotic. Now that you have understood the basic functions of the chip, it is your turn. When you are ready to perform the experiment, please look through the microscope and we will move you closer to the chip for the next steps. Welcome to the microfluidic chip and your experiment control center. Before I ask you to start the experiment, allow me to say a few words about the design of the chip, now that we are closer to it. Right in front of you are the main channels and the mother machines I've been talking about. You will see them in action in a few seconds. To the left and right, you can see the channels that carry the media. Much closer to your right, you can see a smaller version of the microfluidic chip, a syringe with our food sample, and two buttons that you can use to control the flow of the medium. Let's start the experiment. Please load the chip with our food sample by grabbing the syringe and moving it towards the chip. Well done. As you can see, our sample is now loaded onto the chip and some of our Salmonella bacteria are trapped in the mother machine so that we can observe them individually. Now we should allow the bacteria to grow and multiply. This requires a medium that supports the growth of the bacteria. A unique feature of our microfluidic design architecture is the junction at the beginning of the channel. Here we can rapidly switch the media supply that reaches the bacterial cells downstream. This is achieved by applying different pressures at the top and bottom so that only one of the two enters the chip. Please push the green button right next to the microchip to inject this first medium. Perfect, this experiment seems to be running smoothly today. I didn't mention it at the beginning, but due to the design of the chip, our Salmonella bacteria are staying in their traps and can only spread forward because the chip is exactly the same height as the bacteria. We can now watch the mother bacteria multiply. The mother bacteria grows at the end of the chamber and divides into children that are pushed forward and eventually leave the chamber. We can now observe the behavior of a single bacterium in detail. Great, let's move on to the most important part of the experiment, seeing if we can use our ampicillin antibiotic to fight the Salmonella bacteria in our sample. Please push the red button right next to you to inject the medium containing our antibiotics, and let's hope for the best. Just as we feared, it doesn't work. The antibiotic kills most of the bacteria. But some of the bacteria remain alive. Some cells within the population have survived the presence of the antibiotic by reducing or even stopping cell growth, thereby becoming resistant to the mode of action of ampicillin antibiotics, which relies on active cell growth. Thus, some of the bacteria found a way to cope with the presence of the antibiotic. Thank you for doing the experiment for us. Let's go back to the lab to discuss the results. Simply step through the portal to return to the lab. Let's wrap up our results. As you have just observed, depending on the media flow inside the chip, the bacteria behave differently. We used ampicillin for the experiment. Ampicillin and penicillin specifically target the bacterial cell wall, which is a structure absent in human cells, which makes them selectively toxic to bacteria. They interfere with the bacterial cell wall synthesis by disrupting the production of peptidoglycan, a key component of the bacterial cell wall, resulting in a weakened cell wall. Bacteria cells become more susceptible to osmotic pressure changes leading to cell lysis and death of the bacterium. As you saw, this antibiotic worked for most of the bacteria in our food sample. They undergone cell death. However, a subpopulation adapted to the presence of the antibiotics and reduces its growth rate, thereby circumventing the required prerequisite, the antibiotic, to kill off the bacterium, namely, active cell growth. The phenotypic heterogeneity led to the fact that not all cells grew the same or at the same rate, so some bacteria survived the presence of the antibiotic. Well, this means that we will face issues with antibiotics resistance in our case. What do you think? How should we proceed in combating the infection of Salmonella? If you got an idea, please reach out to me, I will do the same. Thanks so much for doing this experiment for us.
It seems that you have fulfilled your tasks as a junior researcher with brilliance today. In my humble opinion, future experiments could focus on testing the effects of combining different antibiotics. This approach is particularly promising in the fight against antibiotic-resistant strains. By using a combination of drugs that work through different mechanisms, it's possible to reduce the likelihood that bacteria will develop resistance. Perhaps, based on this approach and the experimental setup you have just used, we will be able to develop antibiotics or mixtures of antibiotics that will protect us against the idiosyncratic behavior of some of these bacteria, thus taking an important step against antibiotic resistance. Thank you for joining us in the world of microbiology. You can take off your headset now.